Well, thanks everybody. What a great conference, and that was a really cool presentation, Sanjay. Thank you. So my name is Sashank Reddy. I'm the Paul Manson Hans-Jürgen Wyss Professor of Plastic Surgery at Johns Hopkins, and I'm a founder of LifeSprout, a company that we created to deliver true tissue regeneration, which we think is going to define the future of facial aesthetics. And we've heard a lot about regeneration today and regenerative solutions. I think we can all agree that this is where the puck is going. Patients want more natural, more durable results without injecting themselves with forever chemicals that are just going to sit there. And the question arises, what do we mean by regeneration and how do we get there? So I would submit that most of what passes for regeneration in our field is not really that. It's mostly collagen stimulation. It's a glorified form of scarring. When I say regeneration, I mean really bringing back fat, vasculature, truly the tissues that constitute the youthful, beautiful facial contour. That's what regeneration means to me. And if that's the goal, how do we get there? I would say that the best solutions, we've seen some of these today, are tissue-derived materials. They're cadaveric materials. And while those can be great in certain outcomes, there's a certain hesitancy, I think, too, to using cadaveric materials for aesthetic purposes, and it's hard to scale those. So we took a fundamentally different approach at LifeSprout. We made it's truly regenerative products that don't require tissues. They're made from chemically defined materials that we're all familiar with, including hyaluronic acid, which we love, and polymeric nanofibers, and the combination of those two allows us to derive these beautiful regenerative outcomes without requiring tissues. And also, we can combine these materials with the patient's own fat and get outcomes that neither fat grafting nor biomaterials on their own can deliver. And this is why, when I show you an electron micrograph here on the left of our materials, we call it the regenerative matrix, that's the core technology, and I show you an electron micrograph of fat from an animal there on the right. I think if I took the labels away, you probably wouldn't know which is which. So we can get this striking biomimetic structure with chemically defined materials. And that structure is not just great in terms of how it feels when we inject these materials into patients, it's what allows the ultimate regenerative outcomes to obtain. Because of this open, porous nature of this material, it allows for cells to interact in a very natural way and triggers the body's natural regenerative responses to give you fat, to give you vasculature, to give you that natural outcome. And so when we put this into animal models, this is in a rodent model, when we come back and look at the injected material, 6 months later, you see this beautiful proliferation of adipocytes. Those are all the green cells there that we've labeled with perilipin. They're great, they're healthy, they look just like the surrounding subcutaneous fat that's completely regenerated. And there's a nice vasculature that supports them as well. And of course, you don't have to take our word for it. We've published on this rigorously in science journals as well as in our clinical journals. And most importantly, we've also advanced this to the clinic, as I'll get to in a moment. I mentioned to you that we can also combine these materials with fats. When we do fat grafting, much of that fat doesn't take, and even the fat that takes, sometimes it doesn't retain its natural structure. We've all clinically experienced this. Well, when you combine these materials with native fat, you get better performance than you do with either material alone. Superior volume retention and much better structure. And so we're really excited about that as we think of larger applications in the face and indeed in the body with these materials. The first instantiation of that technology in a product is a revolutionary dermal filler that we call Lumina that has completed clinical trials in Europe. It was in a split-phase trial with the leading commercial competitor. It did great, hit its primary endpoint with no significant safety events. And we're actually, I'm hopeful, on the doorstep of CE approval. We've submitted all the regulatory materials for that. And so I think this is going to be a product that will be available in Europe, I'm hoping, very soon. These are just some examples from that trial. The product, again, did great. And we want to actually expand this core technology into a suite of products, including a next-generation version of this regenerative injectable that has collagen nanofibers and is even more powerfully regenerative than the original Lumina, as well as I mentioned, combining these materials with the patient's own fat to give durable restoration outcomes. So we're developing a technology for true tissue regeneration on the doorstep, as I say, of CE mark and European approval. And there's opportunities for us for partnering in Europe and other geographies, and as well as for platform expansion. So thank you so much.